Hello everyone, I welcome all of you to our YouTube channel AgriAddict. So today we are very much excited to start our plant breeding series from our online master course Plant Science 2024. So today we are discussing a topic which you all feel difficult to understand, that is self-incompatibility, which is also important for your exam point of view. So the series which we are conducting in this plant breeding will be helpful for all your agriculture related exams. It could be your CUET or it could be your JRF or SRF exam, whatnot. It could be useful for all your agriculture related exams. So don't miss this session. Stick with me until the end of this session so that you will get maximum benefit. So if you like the content which we are providing, please do subscribe to our channel and also share with all your friends so that they'll also get benefited. So let's get started. So before starting, let us have a brief introduction about myself. So this is your mentor, Roopa MN. So I have completed my undergraduation from University of Agricultural Sciences, Bangalore. I have completed my master's in genetics and plant breeding from Indian Agricultural Research Institute, New Delhi. Some of the achievements in my academic years are, I have been awarded with 8 gold medals in my UG, and I have secured an All India rank of 6 during my JRF examination, and I have also cleared ASRB net. I have secured an All India rank of 28 in my SRF exam. So let's get started. So the topic we are discussing today will be pollination control, that is self-incompatibility mechanism. So who is the first person who has identified this self-incompatibility? Is Coyle-Reuter. Coyle-Reuter is the first person who has reported that self-incompatibility exists in the croplands. So what is self-incompatibility? It is nothing but it is a prevention of fusion of your male and fertile male and the female gamete upon self-pollination. So if you go for self-pollination of your fertile male and the female gametes, they'll fail to affect the fertilization. That is, they won't set the seed. This mechanism is known as a self-incompatibility. What are all the mechanisms by which this self-incompatibility can act? Either it might prevent the germination on the stigma itself. Even if it is able to germinate on the stigma, the pollen tube growth may be slow. So that by the time it reaches the flower might be dried and it might shed, or it is arrested at the stylar stage itself, that pollen tube doesn't grow at all after the stylar region. Or even if it is able to reach the ovule and affect the fertilization, the embryo might abort at the very early stage. Embryo will abort at the very early stage. These are all the mechanisms by which this self-incompatibility is acting. So when we look into the interaction between the pollen and the pistil, we can see two kinds of interactions. So we'll see them one by one. So the first kind of interaction that is happening between your pollen and the pistil is complementary type of interaction, or it is also called as a stimulatory kind of interaction. So the second one is a inhibitory or oppositional system. So these are the two different kinds of mechanisms or the interactive mechanisms that are happening between your pollen as well as the pistil. So we'll see what happens in the complementary type. So in complementary type of mechanism, let us say that your plant is S1, S2, that is your female plant, and let us take S3 and S4 as your male plant. So only when they belongs to the different self-incompatibility group. As you can see, there is no allele that is matching in these two plants. So only when these two plants belongs to the different self-incompatibility group, both the plants will produce some of the substance. Let us say this plant is producing A substance and this is producing B substance, which act as a stimulator for each other. That is, they will stimulate the pollen germination only when they belong to different self-incompatibility group. So this kind of system is operative in case of dendrobium. So this is a complementary mechanism of self-incompatibility. That is, there is some kind of stimulatory substance is there to stimulate the pollen germination upon fertilization if they belongs to the different self-incompatibility group. If they belongs to the same self-incompatibility group, let us say S1, S2 is fertilizing the S1, S2. So there is no substance, no stimulation for your pollen to germinate and affect the fertilization. So pollen will remain as it is. It might get dried and it will lose its fertility. So the next mechanism is your inhibitory mechanism or oppositional mechanism. It is just opposite of that. That is, whenever, let us say you are fertilizing S1, S2 and S1, S2. Whenever the two plants or whatever the gamete and your male and the female gamete, if they belongs to the same fertility group or same incompatibility group, then they produces substance. Let us say this produces A substance and this one produces B substance. What happens? A and B will together inhibit the germination, inhibit the germination of pollen. So what happens if they belongs to the different group? Let us see here. That is, you are fertilizing S1, S2 with S1, S3, S4. That is, they belongs to the different group. So there is no inhibitory effect here, so that the pollen can normally germinate and affect the fertilization. Only when they belongs to the same fertility group, there is some inhibitor production. So these are the two kinds of interaction that is happening between your pollen and the pistil. So usually whatever the self-incompatibility we see in crop plants, that is all belongs to this inhibitory kind of interaction. That is, there will be some inhibitors which will be produced when they belongs to the same self-incompatibility group. So this is a most widely used or most widely seen kind of interaction between your pollen and the pistil. Let us next we will see classification of this self-incompatibility. So Lewis has given this classification based on morphology of the crop plants. So he has divided the self-incompatibility into homomorphic and heteromorphic. As the name itself says, there is some difference in the morphology. Homo means same, there is no difference in their morphology. Hetero is different, there is a difference in the morphology. So heteromorphic, we can see distyly, tristyly kind of mechanisms. So in this homomorphic, we will see gametophytic self-incompatibility and sporophytic self-incompatibility. So we will see one by one from heteromorphic self-incompatibility. So heteromorphic. So in this we have distyly kind of flowers, where di means two, style is different length of your style. So what are the two different style length we can see? One is a long style, another one is a small style. So this will be your anther. Okay. So these are the two different types. So this kind of flower where your stigma is higher than anther, it is called as a pink flower. So where stigma is lower than the anther, it is a thrum flower. So this mechanism is controlled by S gene, which is having S and S alleles. So usually S is dominant to yes. So pin is controlled by small s and small s. Thrum is controlled by heterozygous condition, small s and capital S and the small s. So if we go for fertilization, if we look into the fertilization, what happens? So let us take, we will take pin into pin. That is small s into small s. So since they both belong to the same group, no fertility, they will be incompatible. Let us take pin into thrum. So pin will be small s, this will be capital S, small s. So all will behave as small s. Since it is dominant, S is dominant, all will behave as a S. So they are different. They can affect the fertilization, and they leads to the production of the progeny as capital S, smallest, and smallest in 1:1 ratio. So same with the case of thumb into pin. So again, thumb into thumb, which is capital S smallest into capital S smallest, because they all behave as S, they all will behave as S. So they will be incompatible. These two kind of matings will be incompatible. These two will be compatible matings. So this is a distyly condition. We have one more condition, which is a tristyly. That is 3 different style length. That is, it could be short, it could be medium, or it could be long. So it is controlled by 2 genes, S and M. Usually S will be having a masking effect on the M. Wherever S is there, S will express. So let us see if it is a short plant. So the condition should be yes, S or M, M or S, S, M, M, S, S, M, M, wherever. M doesn't matter. So S is in dominant condition, then it will give a short style. Okay. So only when S is in complete recessive condition, M will express either in homozygous condition or in heterozygous condition. It will give a medium style. So when both are in recessive condition, they will give a long style length. So this is a tristyly. So distyly condition is operative in case of primula and sweet potato. It will be operative. So since this kind of variations we doesn't follow or we doesn't see in many of the crop plants, so it is of less importance for our crop breeding program. So next we will see homomorphic self-incompatibility, which is of very much importance to the homomorphic breeding self-incompatibility. So first we have a gametophytic self-incompatibility. So gametophytic means pollen, gamete. So gametophytic self-incompatibility is nothing but it is a kind of self-incompatibility in which the self-incompatibility reaction is controlled by the genotype of the pollen. Genotype of the pollen. So it was first discovered by Easton Mangelsdorf in Nicotiana sandri. Nicotiana sandri. So we will see some of the common features of this gametophytic self-incompatibility. As I'm saying that it is controlled by the genotype of the pollen. Let us see what happens, how the pollen is produced. We know that pollen mother cell will be there, which will undergo meiosis to produce a 4 haploid microspores. So after this step, each microspore will undergo 2 mitosis. The first mitosis, it will produce vegetative cell and the generative cell. Upon the second mitosis, it will produce vegetative cell will remain as it is and the generative cell will give you 2 male gametes. So here, whatever the substance that is controlling this mechanism will be deposited after meiosis, because it is controlled by the genotype of the pollen itself. Means whatever the substance which is inhibiting the self-fertilization is produced by the genotype of the pollen. That means after the pollen has produced, that means after meiosis has happened. And also in this, we can see that at the 2-celled stage itself, the pollens are released by the plants in this mechanism. And also we have that in this we will be seeing a wet stigma surface. We know that pollen hydration is very much needed for the pollen germination to take place. So wet stigma means pollen germination can happen, but it will be slow. Slow germination will take place. Then where does the mechanism will be acting? That is, the pollen will get arrested in the style region. It will germinate, but it will get arrested in the stylar region. So it will be controlled by the S alleles, which are having a, let us take, you will have a S1, S2 plant. So it produces pollen, that is S1 pollen and S2 pollen. What is the constitution of its style? S1, S2. So in style, we can see that S allele will act in a codominant manner. In case of gamete or the pollens, it is no doubt that there will be single allele. That allele will be expressing. That it can be taken as a dominant form. That is, in case of pollen, it is dominant. In case of style, it is codominant. So what happens to the incompatibility mechanism? We will see that. So here they'll say that here we can see the 3 different kinds of incompatibility groups. What are they? Let us take S1, S2, S1, S2 cross. S1, S2 with S2, S3. S1, S2 with S3, S4. So in first case, since let us take this as a female and this as a male. So female will be expressing S1, S2 equally because they are codominant. So either it can produce S1 gamete, pollen, or S2 gamete. So even if S1 goes, there is S1 allele, it will reject. Even if S2 goes, there is S2 allele, it will reject. So this will be completely incompatible reaction. So in case of this S1, let us take this as a female and a male. So this will be expressing S1 and S2 equally because codominant. So this will produce S2, S3. If S2 goes, there is already one S2 allele which is equally expressing, so it will reject this. If S3 goes, there is no matching allele that they will accept this. So here we will get the 50% fertility or for 50% incompatibility. That is, it is a partial incompatibility, you can say. In this system, we have this is a female and male, S1, S2, it is S3 or S4. Let us take S3 is going and fertilizing. So there is no matching allele. So it will fertilize. S4 is going and fertilizing. There is no matching allele. It will also fertilize. It will be completely compatible. That is, in this gametophytic system, we can see 3 different kinds of incompatibility groups. That is a complete incompatibility, partial incompatibility, and complete compatibility group. So next we will see other features like here polyploidy cannot break this self-incompatibility. Why do you think so? Let us take A1, A2, A3 as a triploid plant. So I'll self-fertilize this. Let us take this as a female and male. So male will produce 3 different gametes, that is A1, A2 and A3. So let us take A1 is going and fertilizing this. So already A1 is there, it will reject. So A2 is going, A2 is also there, it will also be rejected. A3 is going, A3 is also there, it will go and reject. That is, even if polyploidy condition is there, it cannot break this self-incompatibility mechanism. So in this we have, this is controlled by 1 gene or 2 gene, 3, 4. We have all different cases, that is, it is monofactorial, bifactorial. This will be multifactorial, we can say that. So in case of papaver, it is controlled by 3 genes. It has been reported. And what else we can see in this? Pseudofertility. So this incompatibility is controlled by SF allele. So if we irradiate the bud or a pollen, with X-ray or gamma ray, the pollen is able to affect the fertilization upon self-pollination. This is called as a pseudo self-fertility. It is believed that it is because of the allele SF. So this is one more thing that is there in the gametophytic self-incompatibility. And also in this, we cannot see the homozygotes. In this mechanism, homozygotes are not seen. Why? Because let us take S1, S2 crossed with S1, S3. So let us take this as a female and a male. This will produce as S1 and S3. So if it is going and fertilizing this, it won't accept at all. If it has affected the fertilization, then only we could have got this homozygous conditions. So since this plant won't accept this self pollen, which is having a same self-incompatibility allele, so we cannot expect the homozygotes in this gametophytic system of self-incompatibility. And also there is no reciprocal cross difference. Let us take S1, S3 with S3, S4. So let us take this as a female and male. So it would be partially compatible, right? So S3 will match with this, so it is not possible to fertilize. S4 is not matching, so it can able to fertilize. So let us take the reciprocal cross of this S3 and S4 with S1, S3. This is a female and this is a male. So here also S1 is not matching with any of the allele, so it can able to affect the fertilization. S3 has a matching allele, so it cannot affect the fertilization here. So this is a gametophytic system of self-incompatibility. We have a bifactorial gametophytic system, which is not that important for your exam. So in this, it is controlled by two genes. Two genes together control the self-incompatibility mechanism. It is almost very much similar to your sporophytic self-incompatibility system. So today we will be stopping this session here. If you like this content, please do like and subscribe our channel. So we will be coming with the next session, that is a sporophytic self-incompatibility. We will deal with that very quickly. Thank you.